In this video, we describe how to create a TMA and make a block using GALILEO TMAtic, a fully automated tissue microarray. Tissue microarray, or TMA, combines tens to hundreds of paraffin-embedded tissue specimens into a single paraffin block, allowing the simultaneous analysis of molecular targets at DNA, mRNA, and protein levels under identical and standardized conditions on a single glass slide. TMA is an effective tool for high-throughput molecular analysis of tissues, facilitating the identification of new diagnostic and prognostic targets in cancers. It is also a tool in basic research, prognostic oncology, and drug discovery. It provides maximal tissue preservation and use, considering probable limited and irreplaceable archival tissue samples. A TMA project starts by selecting a series of tissue blocks from archives which respond to research biological questions. This must be listed in an Excel file or LIMS, together with a minimum clinical data set for each tissue block. From the identified and archived tissue blocks, a glass slide is made and stained with H&E. The pathologist then can analyze the stained tissue slide using either a microscope or a digital scanned image, or else run the H&E slide through a digital scanner and allow the whole slide to be analyzed using a visual imaging software based on artificial intelligence. In this case, the artificial intelligence selects the area of interest, marks the core areas that should be extracted, and overlays the image of the slide with the image of the tissue on the tissue block. The marked image of each H&E slides and respective extraction coordinates are imported in the GALILEO TMAtic construction folder and used for the construction of the TMA block. At this point, having available all the donor blocks and corresponding tissue slides, we are ready to make a TMA block with the GALILEO TMAtic fully automated tissue array. First of all, insert the recipient and donor cassettes into the 6-position holder and then place the holder on the automatic stage. One of the unique features of GALILEO TMAtic is to be able to work with different types of tissue cassettes simultaneously, such as standard tissue cassettes for all types of archived tissues, macro, used especially for prostate cancer, 96 and 384 microfuge plates for molecular analysis of tissues extracted near the respective core on the TMA block. This facilitates the comparison of the protein and molecular analysis of the cells from the same tissue area. The needle TMAtic checks if the punching needles are bended or broken. The needle integrity check is active every time the head mechanism rotates. This control is exclusive to the TMAtic platform and ensures that the end of the automatic punching cycle is not damaged by a bended needle or incomplete by a broken needle. After checking the integrity of the needles, the barcodes of all block holders and tissue cassettes are captured and displayed on the screen. This check is very important since it controls that all tissue blocks are part of the TMA project and part of the design. TMAtic starts the automatic punching cycle. The first operation is to measure the height of all tissue blocks on the table, including the macro and microfuge plates. The first step is to clean the recipient needle and check that it is empty from residual paraffin. Position the recipient block in the correct position. Makes the hole in the recipient block. Moves the needle's mechanism and cleans the donor needle. Then the donor needle gets cleaned before extracting the tissue core from the donor block. If the stylet mechanism detects that the tissue core is not in the needle, the tissue extraction is repeated, increasing the stylet compression force. This control is exclusive of the GALILEO TMAtic, ensuring that all cores are extracted and placed into the recipient block. In case a microtractor plate is used as a recipient, Galileo will pick and place the tissue core into the selected tube and then cleans the recipient needle. Then the automatic coring cycle will continue until all the tissue cores selected are extracted and placed into the recipient blocks, which can be either the TMA block and or a 96 or 384 microfuge plate. Thanks to the open characteristics of Galileo TMAtic, it is possible, using a robotic arm, to take the macrofuge plate and place it onto an automatic liquid handling system, Hamilton, from which several processes can be made, such as reagents aliquoting, DNA or RNA extraction, capping and decapping, tubes and plates centrifugation, and NGS reaction preparation. The robotic arm can transfer the microtitle plate into an incubator or to a visual image platform like Operetta. As you can see, the Galileo TMAtic platform allows to make a TMA block, giving the opportunity to analyze hundreds of tissue cores simultaneously under the same conditions and make a molecular and genetic analysis of each extracted tissue core from the same block and near the tissue core placed into the TMA block. Thank you for your attention. We hope that this video was informative on how to plan a TMA project, construct a TMA block, and how to analyze the spots. Please feel free to contact us for additional information or clarification.